My name is Pranjal Nautiyal. I'm a PhD student in Florida International University. I work on Thrust Area 2 of CELMET in Arvind Agarwal's lab at Florida International University. My research expertise is in the area of mechanical characterization of materials, interesting architectures, and biological samples. As a part of CELMET, I work closely with Thrust 2 and 3 to understand the mechanical behavior of scaffold structures that are used to support the tissues and the mechanical response of the cardiac tissues. In this presentation, I'm going to introduce the technology that I use to understand the mechanical properties of these materials, some of the key advantages of these techniques, some limitations and how we can overcome those limitations around the use of the mechanical characterization techniques, some key applications in cardiac tissue engineering, and overall expectations from my research to help CELMET project. There are three key aspects where mechanics plays a vital role in bioengineering. First is the mechanics of complex scaffold architectures that are being designed to support cardiac tissues. The three images on top resolve different length scales of the same scaffold. It is important we understand how these hierarchical structures bear mechanical loads. Second, new materials are being explored and developed for printing scaffolds with desirable mechanical stiffness. Understanding the material properties is important before we use them for printing scaffolds. Third, mechanical measurements of cardiac tissues can provide important cues about the effect of different physiochemical treatments or stimuli on the tissues. Here I show the techniques we are using for probing the mechanics of materials, structures, and tissues. Considering the hierarchical architecture of scaffolds, we have adopted a multi-scale characterization approach. We probe the mechanical response of individual features in a scaffold, such as arms and junctions, as well as the overall bulk response. The two images on top illustrate this approach. The fine features in scaffolds are few nanometers thin. In order to resolve these features, we perform in situ testing inside a high-resolution scanning electron microscope. During in situ testing, the deformation phenomena are captured in real time as the structure is subjected to mechanical loading. A mechanical stage is shown in the slide. This stage can be installed inside the microscope for performing in situ tests. Some samples are sensitive to the environment. We probe the mechanical properties of such samples when they are submerged in liquid media. For example, tissues tend to stiffen in dry state as compared to moist state. We can test their properties in moist state by indentation using a fluid cell probe. The lower right image shows the indent imprint in a rat's heart wall captured using this approach. In this slide, you can see mechanical response of honeycomb scaffold is being probed at two different length scales. These tests were performed inside electron microscope and deformation was observed in real time. The image and curve on top shows the bulk compression of honeycomb structure. The bottom result shows highly localized indentation using a sharp pyramidal tip, where a localized feature in the scaffold is being tested. So we can extract visual information about deformation as well as quantitative data from in situ testing techniques. It is also important to know what happens when scaffold structures are subjected to multiple loading-unloading cycles. We are using in situ characterization approach to understand time-dependent mechanics of the materials and architectures used for supporting the cardiac tissues. The video here shows cyclic deformation of a porous architecture by in situ indentation. There are some challenges associated with mechanical characterization which require attention. Although in situ approach is highly informative, one major limitation is that it's time consuming for installing the samples inside the electron microscope. It can become expensive due to the instrumentation involved. The time and cost issues can be addressed by improvising the design of in situ stages such that multiple samples can be loaded for higher efficiency. Another major challenge is indentation testing of cardiac tissues. These tissues are extremely soft, making accurate determination of mechanical properties difficult. It is important that environmental noise is minimized by creating effective isolation. Probe or indenter tip selection is also critical for testing soft samples. Large, blunt tips should be used to avoid excessive stress concentration. So, if we look at our three-plane diagram, this work aims to develop desirable metamaterials by evaluating the mechanics of new materials and architectures. Later, these metamaterials will be integrated in the test bed. I'm going to highlight two key applications of the in situ characterization technique in CellMet. First, in situ tests provide feedback for effective design of scaffolds with desirable mechanical attributes. The images in the top row show different architectures that can be designed to cater different support requirements. Second application is the development of micro-tissue gauges or platforms for engineering cardiac tissues. The bottom row shows the PDMS pillars in a tissue platform that undergo bending due to tissue contraction. In situ testing is being used to measure the stiffness of these pillars. Our goal is to develop a database of mechanical properties of the materials used for making scaffolds. We would like to establish correlation between printing conditions and the scaffold properties, and our prime motivation is always to understand how different scaffold architectures used for cardiac tissues respond to mechanical loading.
This is a field emission scanning electron microscope. We perform in situ mechanical testing of different materials and structures inside that microscope. We use nano-indentation approach. This is an indentation stage. This stage has a sample holder where we can mount the samples that we want to test. We have a tip holder here which can hold different kinds of tips that we use to probe the mechanical response of the material. The stage can be installed inside this microscope chamber so that we can observe the real-time deformation videos as the test is being performed. We can see the deformation in this monitor screen. So for example, right now you see a honeycomb scaffold structure which is being indented inside the SEM. So in situ mechanical characterization is a very powerful approach because you're able to extract load displacement characteristics while simultaneously looking at how the material is deforming in real time. Now let me show you different parts of our in situ indentation stage. This is an in situ nano indenter. This is the transducer which controls the loads applied during nano indentation. Here you see a tip holder where we mount different kinds of tips to probe or obtain the mechanical response from the sample. This is the sample holder. We mount our samples here. Right now, a micro lattice is installed on this holder. During testing, the holder part is brought closer to the tip for performing the mechanical test.
Thank you.